Okay. After that, I'd like to invite Dr. Walia for his talk. Dr. Walia is a clinical geneticist and full professor at Queen's University in the Department of Pediatrics. He has published about several rare genetic disorders in multiple peer-reviewed manuscripts. He is a member of the Ontario Genetics Secretariat Committee that is focused on developing a provincial strategy for genetic services and practice in Ontario.
Good afternoon, everybody. First of all, I want to say thank you for the ACD to invite me here and ask me to give a speech. Second, thank you more because they are accommodating me to give me a talk today when I could not come yesterday. And third thing, very, very thankful that they have put me in front of lunchtime so that now you guys are hungry and I'm making you sit here. So I am really sorry for that. So what I wanted to talk about was that what is the, if there is going to be a future clinical gene therapy, which I think this talk is going to be more for parents, that what the parents should know. I know there are many physicians, many researchers, but I will talk about very basic stuff that I think all the parents should know. Thank you. She always points me in the right direction. So I have some disclosures that I sit on advisory board for certain industries, and I get some funding from different agencies as well. So my goal after this is that after this presentation, the participants or the parents will be able to list at least 3 possible complications of gene therapy. What are the advantages? But there are we'll also list at least 3 concerns with gene therapy. We'll discuss that how it goes from bench side in labs to bedside. So it's almost like making a child. The projects of a gene therapy is like making a child. It starts right from the idea stage, and then from idea it goes into making the child, and then it slowly progresses to a mature form where it can be of some use for other people. So similarly, same thing is here, that if you see, it goes from the idea stage where there is, you want to treat a disease, and then it goes into a long process of preclinical development, and that means there is a proof of concept that actually your idea will work to treat. And then, and most of the proof of concept means you're going to take your idea into cells, into organoids, into animals, and once you say, yeah, it's going to work in animals, then it starts getting into optimization. Optimization that, for example, Laura Berencelli was saying that, oh, I tried in mice died. So that means the concept is not working because there are some complications. So once it has to be taken from the idea to the point where it's working and it's safe to progress further. And then you start making, how are you going to deliver the treatment? Is it going to be through IV? Are you going to do it intrathecal? Are you going to do it intraperitoneal? We don't know. And then the dosage, as she said, high dose can kill mice, but you have to find the right dose before it goes into humans. Then the most important part are their immune responses, because these proteins, the children may have not seen it before. So when you are entering into an area of immune system, you need to make sure that your immune system doesn't kill those proteins. And then, is it toxic? You have to prove it in animals before, large animals, and then it goes into humans, into clinical trials. So let me start with how the inspiration comes into ideas. It's basically from the parents, and they are the drivers of many researchers going into the clinical trials, and that's what you guys are doing. You are bringing all the researchers and clinicians together. Basically, a child usually sometimes is born normal, sometimes not normal. You get sometimes many loss of skills, like hypotonia, ADHD, different diagnoses. And That's how the parents become so desperate that I need some treatment for my child. This was just a story of one of my patients, and that's how I started working on one condition for that family. Anyway, so what is actually a gene therapy? And I'll give you a very brief definition. What is happening is you're going to introduce using a vector. Vector means it's like a car, and you're going to put the treating person in the car, and those are nucleic acids, and the whole idea is that you're going to alter the gene expression. Since it's a genetic condition, so you want to alter it. And the alteration can be, you want to increase it, decrease it, or halt it. But the whole idea is we want to alter the pathological disease, whether by preventing it or halting it or reversing it. So what will happen is there will be, for our gene therapies to work especially with the viruses, there will be a viral vector. It needs to go into the cell. Into the cells, it needs to go into a special area of the cell called nucleus, where your genetic material is lying. So the genetic material gets from here, gets translated into proteins in a special machinery. So the virus has to come from there, get to the cell, into the genetic material, whether it stays outside in that compartment or goes and integrate into the actual genetic material, which can keep going. That is important. And then from there, the protein is made, and then it gets expressed and it does its job. So there's a whole process of the machinery which needs to be taken care of. And there are many differences, possibilities that can go wrong. So a lot of optimization needed. How it can be delivered? It can be just a gene addition. So by gene therapy, you're just adding a gene. So when you heard of some previous trials with AAV, you're adding a gene. That means the whole entire gene is getting added into the system. You're trying to supply the protein when it's made. Or there is a mutation, which is a common mutation, that you're going to correct it. That is gene editing. So that can be done for gene therapy. You don't need to add the whole gene, but you're just correcting an existing mutation. And then there is gene knockdown. And the gene knockdown means you're making too much, for example, AGAT gene, and if you knock it down, maybe you will not make GAA. So that's kind of treatment. I know you all are hungry. I don't understand that. But I'm just hold on for some more seconds. So just showing you how it can be given. It can be given by, you take the cells out of the body, modify the cells, put it back. That may correct some of the CNS diseases. You may inject into the blood, into the spine, or directly into the brain. So you have to choose which way you're going to give the treatment. And there are different advantages or disadvantages of all of them. So then comes, when somebody is testing it, what you want to see is that there is always this downward trend where there is disease, but whenever you want to treat, this green line gives a very good hope. That means you're correcting it. When you see that line, you really, or you want to see that you have improved the survival. And that's what the proof of concept is. And that gives you some happiness, right? So those happiness are the moments which you are looking for, but then there is more work which is getting created. When you create more work means that whether the gene expression is going down with time. So the gene expression is going down means initially you show good gene expression, but in long term, the gene is going down in systems. So you have to make sure that whatever treatment you give expresses and stays there for a long time. So then is immune response, is very important, which parents need to know, and it is very important. Immune system is at your service in normal human body, and is always watching, is there anybody new coming in the field of territory, which they have to kill or take care of. And then whenever they see something new, there are two types of cells which are always in action. There are B cells who can throw weapons far, and they are called antibodies. And there are T cells which can just come in contact and kill those foreign materials. So those are the two cells which the immune responses, whenever you give gene therapy and you make the protein, it may be a brand new protein for that child, and the immune system can just wipe it out right away. And that's very important that for any gene therapy, you need to be sure that that is being taken care of. And that is why many of the gene therapy trials right now, they are using immunosuppression systems. So whenever the trial will start, you will see there will be an immune suppressive regimen always given to a child along with gene therapy. So in our trials where we are running some trials, we are giving rapamycin and prednisone, but there are many more immunosuppressive regimens being used by different groups. That can include immunoglobulins, different, some kind of drugs which can actually kill temporarily some B cells, so different combinations. So in our group, what we did is we saw that when we use a combination, we can suppress the immune system quite well, and it really worked, not only for B cells, but also for T cells. Then the route and dosage, which need to be seen that which works. For example, I'm just giving you just example for another disease, that if you give a lower dose, the response can be very small, but you have to optimize the dose so that it becomes a response which is equivalent to a normal person. So dosage has to be calculated, and then you have to see that it is not toxic at the same time. All right, so toxicology is very important, and that's what Laura Baroncelli is looking at. But in mice or animals, what we are going to look at is that we choose a dose, and then we show that there is no difference in your blood, in your liver cells, and your vector is still there for a long time. So that's the phase which you're going to go through. And when you put the toxicology study, that's what actually Health Canada or FDA or EMA will look for, and it will be a huge amount of paperwork to set up for clinical trial. When you do a clinical trial, it goes into phase 1, phase 2, and they can cost anywhere from, any idea how many dollars it will cost? $10, $100? No. It'll cost anywhere from $5 million to $30 million to run a phase 1, phase 2 trial. So you need an industry partner at that time. So whenever any researcher, any physician scientist will be ready, either the NIH will just open the tap and the money will flow, or the industry has to open the tap. Somebody has to open the tap. All right. And then there will be processes which will take, it will be showed up in the clinicaltrial.gov website. You have to decide how many months of age you're going to give treatment initially. It'll be less than 15 months, it'll be less than 1 year, it'll be at birth, we don't know. FDA doesn't like to treat children. They want to treat adults first. So who will get treated first? What will be the immunosuppressive regimen? What will be the route? What will be the dose? So all needs to be taken care of. And I have put ACD1 just to show that maybe it's a drug, and ACD is going to be involved here, and it's called ACD101 at that time. So then there will be a timeline. So when the parents will be involved, the ACD101, it'll be given, but that there will be a time when they'll be held to see that the first child is doing well enough. That's called first in human clinical trial. Once the child is doing well for at least, let's say, 2 months or 3 months, then you're allowed to inject second patient. And then it depends on the regulatory agency, whether this is going to be FDA or Health Canada. you can keep proceeding to patient 2, 3, 4. And there will be a huge board looking at your data. It's called Data Safety Monitoring Board, who will tell, yeah, you can go to second patient, you can go to third patient or fourth patient. So lots of stuff going on. So whenever you're going to make a trial, and I think that there was a session yesterday, they looked at what is required for a clinical trial. You need biomarkers. There's a huge list of primary markers, secondary outmarkers, and exploratory biomarkers, which need to be explored and tabulated before that. And the good thing is that we are already working on this. So thank you for the association, and thanks for the members here. And then the complications, what can happen? In the first few weeks when your gene therapy will come, that's the most critical period because then body reacts right away. It's possible that there may be some immune reaction, side effects, acute effects. But first few weeks from 2 weeks to 6 weeks is very important when liver, especially if you're going to use AAV, the liver will get kind of not happy, so irritated. When the liver is irritated, the liver enzymes will go up, and that's what you're going to monitor. There may be blood cells which may go up and down. That means your hemoglobin, the fighting cells, it's called neutrophils, or all those things can go up and down. The immunosuppression can make you even the child very susceptible for infections. So all those things need to be taken care of at that time. So parents may have all those questions, and whenever you will sign the informed consent form, you will read all those possible complications, and you will sign and say, I am ready to take all those risks. And then only your child will be able to participate in trial. And then there will be an ordeal of months to years, and then it'll be followed for, some trials followed for 2 years, some trials followed for 5 years. So there will be a lot of things. We don't know anything. We know about some complications. For example, it can affect liver, it can go into brain, but there are possible new complications. Like for a trial for Duchenne muscular dystrophy, a new complication like called thrombotic microangiopathy showed up, which nobody knew before, and then it showed up. So there may be many new complications which will just keep popping up at that time as well. And then the copy numbers, how long is your gene therapy going to survive? You have one chance at present to get the gene therapy because the field has not yet figured out, can we give gene therapy again? So it's a one-time shot, and you want to make sure that whatever cells gene therapy you get, it just persists for a very long time. So there are a lot of stresses which are there not only for families, but also for physicians, for researchers, especially emotional. We have seen some patients who receive gene therapy in our clinical trial, and every day there is something new, and they're always on call with us because you don't know what's going on with your child. You don't know that she vomited today, is it because of gene therapy or there's a new bug? So there's a lot of going to be positive and negative emotional levels at that time. The positive part is that it's getting treated. It may be a one-time treatment, and your child may get treated for a very long time. Some parents have high hopes that their child may get cured, but we don't know yet. Financially, it may be problematic because you may get the treatment at your home state, hometown, but you may have to travel across the sea. And if you go there, and if both of your earning partners go, there may be a loss of income. So who will pay for it? Sometimes the industry may pay, sometimes industry doesn't pay. So just make sure that that is a possibility. Who will cover the insurance? So your child may be covered with the insurance, but if you are going with the child and you break your bone, somebody has to cover your recovery as well. So there can be stresses for that. Time commitment. If you have 3 more children at home, you are treating one child, how are you going to take care of that other 3 children? So And then fear of unknown. We don't know that what's going to happen after treatment. Nobody knows because it's still a research at that time. Industry commitment and funds. Sometimes industry is really good company like Pfizer or Novartis. Sometimes it's a new startup. The funding may just disappear. Biotechs are going through a very hard time. And they may decide to say, your program is no more priority for us. It's another stressor. And then regulators, they want more data, more safety pauses, so there can be a lot of hiccups as well. But all these stresses are okay to take the risk because it's ultimately your child. You may get cured, your child may get cured. And that's what we are looking for, right? The researchers, physicians, what we are striving for is that for one hope, that the child may get treated. Patient first. So there are issues with genetic therapies, and that includes gene therapy. It can go randomly anywhere right now. AAV is still said that it stays outside our main genetic material, but 5 to 10% of the times it does integrate, and we don't know what it's going to do in long term. Cancer? No, cancer, we don't know yet. Cost after commercialization, the SMA treatment at present is costed around $2 to $2.5 million per dose. So even if it goes into commercial, there will be a cost. Hopefully the insurance companies will pay. Sometimes they don't. When it goes into, and there are ethical questions, you give the gene therapy, it may go into change your sperms or eggs. So we don't know what's going to happen in the next generations. At present, we don't know. Some people just say that you are changing the course of a normal disease and you're trying to play like a god, and some people don't like that. So it's a long journey. It's like my Delta flight yesterday, day before yesterday. It was supposed to be a 6-hour journey, but it took almost 36 hours. So it can be a journey for any project, and you start anticipating, oh, it's going to take small time, maybe 1 year, but it may take 10 years. We don't know what's going to happen. So it's a long journey which will involve parents, the child, the whole family and friends as well, researchers, industry, regulators, government. But that's all is going to happen in any gene therapy trial. And I thought that I should tell the parents that there are it's going to be a journey. And I am sure that we are all working towards it. I'm working towards it. I'm sure that we both can work together, and we all will find someday a cure for our children. I'm done.
Thank you so much, Dr. Walia. That was a great presentation. Thanks for going over all of those concepts with us. You made it so that I don't know if we have a lot of questions.
Lunch is ready.
That's right. So we have 5 minutes. If someone has questions for any of the speakers, I'm happy to walk to you.
Sorry, I have lots of questions. But just for this one, so we've heard a little bit about antibodies after for the viral vectors and about only getting kind of one-time shot for gene therapy. So how does the COVID vaccinations affect our ability to get the genetic, to get treatment, later gene treatment?
So we are getting vaccinated by injections or by infections all the time. So whether you take immunization or you go and get some bug from anywhere, your antibodies are made every day for something. So if you have got COVID and you got immunization, that is not affecting for any future for your gene therapy. No, but it doesn't matter. So for AAV9, if you're getting a bug with AAV9 and you're seropositive already, previous many trials have stopped you from being enrolled because you have already some antibodies against it. if it's an AAV-induced infection previously. But if you're giving treatment in immunoprivileged areas with immunosuppression, they are not stopping you to enroll, whether you're seropositive or seronegative. That means if you're giving injection intrathecally or into the brain or intracisterna magna, you're okay.
Hi, this is Kristen Voorhees with Ultragenics. This is more of a comment just about the term cure. I think we need to be incredibly careful about that word in all gene therapy. Our company is working across a number of programs and diseases for that. And across industry, we're really careful about that term because we don't know what the durability of gene therapy is in general. So I would just caution against using that in the context of the presentation. Thank
You're
you.
absolutely right. So when I say cure, the reason I use the term cure means that parents are hoping for it, but it may not be a cure. It may not work at all. I have 3 patients already enrolled in multiple. My own patient went into some other state, got gene therapy trial somewhere else, came back, didn't work. So there is a possibility that if you give gene therapy in the first few phases, if it's given a low dose or medium dose, it may not work at all because it's still a research for finding the right dosage in humans. And it may not work, it may work for small time, it may just stabilize the disease. We don't know what it's going to do to your child initially. It's a research. Initially, many physicians treat the patients as if they are just subjects, as if they are numbers. And I have heard it from many physicians saying to those enrolled patients and say, look, I want to be very clear, this is research. I'm just putting a number to this, to your child because it's research. I understand your patient, and there are a lot of emotions attached to it, but I don't know it's going to work. There will be a full discussion with any of the family if their child is getting enrolled with in a trial, because it is still a research trial.
I would like to add to this. I understand the industry has to be very careful and cannot make any inappropriate promises. So this is why using this word cure from industry is a bit different than when we as physicians use this word cure or curative, right? We don't necessarily mean that this is forever, but it is different to an improvement. You know, most of our treatments are not a cure because we can just mitigate some of the problems, while gene therapy under certain circumstances could be a cure, even if it doesn't last forever. So this is why I continue using this word cure in contrast to treatment, improvement, or so, just to make the difference. And I think for the families, for our patients, this is very important because it is a very significant difference. Whether what we try is providing a cure or an improvement is a different question. But I think we should continue using this language when we talk to our patients and families.
Let's make it a compromise between industry and physicians. Can it be a partial cure, mild cure, moderate cure, long-term cure? We don't know. Is it good? No, you're right. Even in cancer.
Not just industry, it's physicians and communities and other patient organizations and disease states have been commenting on the importance of language.
You are absolutely right.
So that's really just the direction of where the comment is coming from.
I understand, because when you say cure, it raises the hope of parents, and that's what people are trying to be cautious about. That don't raise the hope of parents that much by using the word cure. But we are using a lot of, like in oncology, we are using the word survival or stabilizing the disease. So there are a lot of people use different terms so that explains it appropriately. But as physicians, we have our hearts attached to our patients. So we do take, it may be a very mild treatment. It may not work. It may work, it's called partial treatment, or it may be moderate, it may be long-term cure, it can be long-term treatment. And I agree with actually Andreas. There is more heart-to-heart connection there by saying cure, because, and even if you say it can be a partial cure, but it's a treatment. So to give a hope when there is no hope.